Hello guys, welcome to my channel. So we are continuing with the protein function. So we discussed about immunoglobulin, myoglobin, then actin and myosin. Now we'll discuss about the most important in the protein, that is enzymes. So we'll study about the enzyme. So the first thing we'll discuss about the introduction of the enzyme. All right, so there are two fundamental conditions for life. The first is the organism must be able to self replicate, right? Like the cells. Second is it must be able to catalyze chemical reaction efficiently and selectively. So the central importance of catalysis may seem surprising, but it is easy to demonstrate. So, as we know, living systems make use of energy from the environment, right? And many of us, for example, we can see like consume substantial amounts of sucrose, that is a common table sugar, as a kind of fuel, usually in the form of sweetened foods and drinks, correct? So the conversion of sucrose to CO2 and the H2O in the presence of oxygen is a highly exergonic process, releasing free energy that we can use to think, move, taste, and see, right? So the energy that is converted, that is used when we think, when we move, when we see, right? So, but however, when you see a bag of sugar, right? A bag of sugar that can remain on the shelf for years without any obvious conversion to the CO2 and H2O. Although this chemical process is thermodynamically favorable, but it is very slow. So that's why you don't know, like when it's the sugar is still sitting on a shelf, like it will be just there. It won't like suddenly be converting into a CO2 and H2O. But yet this when sucrose is consumed by human, it releases its energy in seconds. How? The difference is catalysis, right? Without the catalysis, chemical reactions such as sucrose oxidation could not occur on a useful time scale and thus could not sustain life. All right, so why we discussed about catalysis? Because enzymes. So enzymes, the most remarkable and highly specialized proteins, and enzymes have an extraordinary catalytic power, often far greater than that of a synthetic or an inorganic catalyst. Right? So they have a high degree of specificity for their substrates. They accelerate chemical reactions tremendously and they function in an aqueous solution under very mild condition of temperature and pH. Right? So that's why enzymes are the central to every biochemical process. Right? Acting in organized sequence, they catalyze hundreds of stepwise reactions that degrade nutrient molecules, conserve and transform chemical energy, and make biological macromolecules from simple precursor. So these are the enzymes, and that's why the study of enzyme has immense practical importance. Like for example, in some disease, especially inheritance genetic disorders, there may be deficiency or even a total absence of one or more enzyme. So that causes that inherent or genetic disorders. In other disease conditions may be caused by excessive activity of an enzyme. Measurement of activities of enzymes in the blood plasma, erythrocytes, or tissue samples are important in diagnosis certain illness as well. And many drugs act through interaction with the enzymes as well. And enzymes are also important practical tools in chemical engineering, food technology, and agriculture. Right, so that's why enzymes are very important. Right, so we'll just talk a little bit about the history of enzymes. So much of the history of biochemistry is the history of enzyme research. Biological catalysis was first recognized and described actually in late 1700s in the study of digestion of meat by secretions of the stomach. But like research continued in the 1800s with the examination of conversion of starch to sugar by saliva and various plant extract. And in 1850s, Louis Pasteur concluded that fermentation of sugar into alcohol by yeast is catalyzed by ferments. This was in 1850s. Uh, but then in 1897, Edward Buchner discovered that the cell-free yeast extract could ferment sugar to alcohol, providing that fermentation was promoted by molecules that continued to function when removed from the cell. So Buchner experiment marked the end of vitalistic notions and dawn of the science of biochemistry. And then after that, Frederick Cohen later gave the name enzymes to the molecule that were detected by Buchner. So this is that's the history of enzymes. How it came then, like the isolation and crystallization of ureas by James Sumner in 1926 was a breakthrough in early enzyme study. And the Sumner found that urea's crystals consisted entirely of protein, and he postulated that all enzymes were proteins. But like at that time, like there was like a controversy regarding that, but like in 1930s, Sumner conclusion widely accepted. Why? Because after John Northrop and Moses Kunitz crystallized pepsin, trypsin and other digestive enzymes and found them also to be protein. So this was some of the history of enzymes, like from 1700s. Research are going on to find and research about the enzymes, and since the later part of the 20th century, thousands of enzymes have been purified, their structures elucidated, and their mechanisms explained. Right, but most enzymes are protein, right? Not all of them. Most enzymes are protein. Most enzymes are protein, with the exception of few classes of catalytic RNA molecule that are not protein. All enzymes are proteins. So their catalytic activity depends on the integrity of their native protein conformation. Remember that. Yeah, the catalytic activity of enzyme depends on the native conformation, right? If an enzyme is denatured or dissociated into its subunits, catalytic activity is usually lost. And if an enzyme is broken down into its components, amino acids, its catalytic activity is always destroyed. So thus the primary, secondary, tertiary, and quaternary structures of protein enzymes are essential to their catalytic activity, that are very important. Right, so enzymes like other proteins have molecular weights ranging from about 12,000 to more than a million. So some enzymes require no chemical groups for activity other than their amino acid residues that they have, right? But others requires an additional chemical component that is called a cofactor, right? So some enzyme can work alone, but some enzyme requires a chemical components, right? So that is called a cofactor. Enzymes require a chemical component that is called cofactor, right? That's called cofactor. So either in the cofactor, that is either one or more inorganic ions. Like what are the cofactors? Like such as Fe2 plus, right? Magnesium or zinc. Right, so these are some of the cofactors, or like if not cofactors, then there is a complex organic or metallo-organic molecules also can be attached with enzyme that are called coenzymes, right? The additional instead of a chemical component, there is a complex organic or metallo-organic molecules, right? Instead of this is attached with our complex organic ions, right? Organic or metallo-organic molecule. organic molecule. That is called a coenzyme. That is called a coenzyme, right? If it's attached with this, it's cofactor. If it's attached with a complex organic or metallo-organic molecule, that is called coenzymes. So coenzymes act as a transient carrier of specific functional group. So what's the function of coenzyme? They act as a transient carrier of specific functional groups. And where these coenzymes are derived? So they are derived from vitamins, organic nutrients that are required in small amounts in the diet, right? So some enzymes require both a coenzyme and one or more metal ions for activity, right? They require either a coenzyme or one or more metal ions for activity. A coenzyme or metal ion that is very tightly or even covalently bonded to the enzyme protein is called a prosthetic group, right? A coenzyme or metal ion that is very tightly or even covalently linked, that is bound to an enzyme protein, is called a prosthetic group. A complete catalytical active enzyme together with its bound coenzyme or metal ions is called a holoenzyme, and the protein part of such an enzyme is called a apoenzyme or apoprotein, right? Right, so I'll write this down. So a complete catalytically active enzyme that is together with its bound coenzyme, right? It's bound coenzyme or metal ions is called and a holoenzyme, right? That is called a holoenzyme. And the protein part of such enzyme that is called apoenzyme or apoprotein, right? Right, on your screen you can see the table that shows some inorganic ions like this that serves as a cofactor for enzyme, right? As you can see, the Fe2 plus, that is a cytochrome oxidase, catalase or peroxidase, that are the enzyme that is with the Fe2 plus, or you can see potassium, that is pyruvate kinase, right? So that are some of the example you can see in the table. And the another table that you will see that is shows some coenzyme that serves as a transient carriers, as I mentioned, of specific atoms or functional groups, right? As you can see, that's coenzyme biotin. What it transfers, it transfers carbon dioxide, and the dietary precursor is biotin, right? The most common is, you can see, the vitamin B12, and the coenzyme is 5-deoxyadenosine-cobalamin. That's coenzyme B12, right? And it transfers H atoms and alkali group. Right, so and how enzymes are named. So as enzymes are classified by the reaction they catalyze. So many enzymes have been named by adding just the suffix ase, like this, like urease, right? Polymerase, like the suffix ase, and to the name of their substrate or to a word or phrase describing their activity. For example, urease, you can see that catalysis, like example, urease. What it does? It catalyzes hydrolysis of urea. DNA polymerase. What it does? Catalyzes the polymerization of nucleotides to form DNA, right? So there are some enzymes. It's named after what what activity they do. Other enzymes were named by the discovery for a broad functions before the specific reaction catalyzed was known. So, for example, an enzyme known to act in the digestion of food was named pepsin, right? That was from the Greek pepsis. So, example, the pepsin derived from that is derived from Greek pepsis. That means digestion, right? That means digestion, right? This is how it was named pepsin. And lysozyme was named for its ability to lyse, that means break down bacterial cell wall, right? But still there are others were named for their source, like trypsin, that's named in a part of a Greek tyrene to wear down and was obtained by rubbing pancreatic tissues with glycerin. Sometimes the same enzyme has two or more names as well, or two different enzymes have the same name. So, because of such ambiguities, as well as the uh, ever increasing numbers of the newly discovered enzymes, the biochemist, what they they did an international agreement that have adopted a system for naming and classifying enzymes. So this system divides enzyme into 6 classes. What it does, it divides enzyme into 6 classes, right? With each with the subclass based on the type of a reaction catalyzed. So each enzyme is assigned a 4-part classification number and a systematic name which identifies the reaction it catalyzes. As an example, the formal systematic name of enzyme catalyzing the reaction. Alright, on your screen you can see the picture. That's the table of international classification of enzyme. You can see that the number 1 class has oxidoreductases, and the reaction, what it does, it transfers of electrons and so on. You can read all this. Transferase, hydrolases, lyases, isomerases, ligases, right? So these are the enzyme classification. Okay, so that was just an introduction for enzyme. Will not go in deep in this video. So in the next video, we'll study about how enzyme works and more functions for the enzyme. All right. So thank you guys for watching, and we'll see you in the next video. Thank you.